Hello everybody, welcome back to another video for Unit 4. This one's going to be all about cell cycle regulation, which is actually one of my favorite parts of this whole unit. I don't really care all that much about the steps of metaphase or mitosis or interphase. It's important, I'm not saying it's not important, especially for developing our language, but in terms of what is really cool to me, cell cycle regulation is awesome. And so let's go ahead and get started. I asked you guys in class to start by talking through this figure, so I would strongly encourage you to talk through it again, pause the video and talk through it again, but it really focuses on the checkpoints. And depending on the source that you look at, you might find a different number of checkpoints, but the ones that are really, really important are G1, the G2, and the M checkpoints. Now, I'm probably not going to ever ask you specifically what happens at each of these stages, but I do kind of want to talk through it. So at G1, this is before we really even started the cell cycle. So at this checkpoint, the cell is really making sure that it has the correct conditions to replicate. So is there enough space? Is there resources? Is there energy? If not, then the cell is going to go into something called G0. And G0 is just a non-dividing state. That's important that you know that G0 is a non-dividing state, and our cells are in there a lot of the time, and then only when they need to divide is this checkpoint, this barrier, going to go down so that we can proceed through the cell cycle. The next one I want to talk about is this G2 checkpoint. Well, this G2 checkpoint, there's a phase in here that we're not seeing, which is S, and hopefully you remember that S is where DNA is replicated. And so in that, we need to make sure that the DNA was correctly replicated. If it's not, then you can have some really serious problems. And so that's sort of what G2 is responsible for. Now, like I said, I'm never going to ask you specifically about any checkpoints because there's sometimes a lot of overlap, but in the G2 checkpoint, you can think of it sort of as making sure that the DNA was correctly replicated. G2 also is at the end of G2, and so you might argue that you got to make sure that other things are replicated correctly as well, some important organelles. So G2, if it's not replicated correctly, this is the problem. We can't just go into a state like G0 because we're no longer just a normal cell. And so realistically, what's going to happen here usually is the cell's going to say, hey, let's die. That's called apoptosis. I'm going to write that to the side. It's a very important term that you're going to see throughout the year. But apoptosis. Sometimes you'll hear people say apoptosis, which I find a little weird, but I'm going to accept it just because I have heard it several times. Okay, so apoptosis is basically cell suicide. It's programmed cell death. It's not an accident. This is the cell telling itself to kill itself. This is different than something like necrosis, where if you were to maybe wrap something around your arm really, really, really tight, eventually your hand would die. Not a good thing, but your hand would die and it would turn black, and that's called necrosed tissue. That is different than apoptosis. Apoptosis is designed, it is to actually keep the organism healthy, whereas necrosis is a mistake. It really doesn't want to do that. So that's the option that we have here at G2. It ends up, if things don't go right, it's going to go through apoptosis. Same thing with M. M is in the mitosis phase, and this is going to do a very similar thing. If things aren't going right in mitosis, at the prometaphase stage, then it's going to tell the cell, hey, let's kill ourselves because we are not going to be a healthy cell, and then that could cause problems for the organism. Okay, so those are the checkpoints. This is how our cell cycle is regulated. If the conditions are right, then we get to proceed. If they're not, then we're either going to go into G0 or hopefully apoptosis. And so we talked way back in the cell signaling about a nuclear response to a ligand. And so how do we stimulate the cell cycle? How can we even get the cell cycle to start in the first place? Well, typically it begins with a signal. And these signals are called growth factors or mitogens. They're very well named, right? Growth factors, meaning it's telling the cell to grow. That's what the ligand is doing. Or it's a mitogen, and a mitogen, we've seen this prefix in mitosis, same idea. So it's going to stimulate mitosis. So these are things that can stimulate it, so they are also part of regulation. If they're not there, then the cell's not getting the signal to say, hey, let's keep going, let's divide again. Okay, now what are some things that can stop the cell cycle? So we know what can stimulate it. Those are growth factors and mitogens, but things that can stop it are going to be things that are often in the environment. And so density or high density cells don't want to divide. This is called density dependent inhibition. And so if the cells get too close to each other, they will send a signal saying, hey, we're too close. If we divide again, we are going to overwhelm either the system or some sort of problem will arise. Now, that is high density. They don't want to be too close. Instead, they like to be attached to the extracellular matrix through a bunch of anchorage or anchor proteins. That is not totally what it means by anchorage dependence. Cells do want to anchor to the extracellular matrix, but this also actually goes as well into cell culture. It's very difficult to culture some cells, not all cells. You can culture bacteria oftentimes in liquid, but they really like to have a surface to really stick to. Otherwise, they have a really hard time dividing. Okay, now we're going to start talking about the more exciting, maybe the part that I find more exciting, which is cyclins and CDKs. So I asked you in your book to read page 197, and it describes cyclins. Well, cyclins are proteins. They are very, very important proteins that are largely important for regulating the cell cycle. So how do they work? Well, they work with something called CDKs. Let me write that down. CDKs. Now it stands for cyclin-dependent kinases. Well, we've seen this term kinase before in cell signaling. We've seen it all throughout the unit. Kinases phosphorylate. And so you might imagine that when a cyclin activates the CDK, it is going to trigger a phosphorylation cascade, typically. Now, this is a pretty nicely named thing as well, right? Cyclin-dependent kinase. So it tells you that this CDK, cyclin-dependent kinase, is dependent on a cyclin. So if the appropriate cyclin is there, it is going to activate a specific CDK. And that CDK, because it's a kinase that is now active, kinases phosphorylate, so it'll trigger a phosphorylation cascade that will ultimately lead to a cellular response. This is how we regulate a lot of the cell cycle. Pretty exciting. So let's look at that in a little more detail by looking at these figures. The first one here on the left is showing you how we have a change in cyclin concentration over time. This is pretty important because it really tells us how we trigger one phase to the next. So we have the G1 cyclin, G1S cyclin, the S cyclin, the M cyclin, and they all peak at a certain spot, and they often trigger the next phase. That's pretty cool. The nice thing is we really only need to have one of the two proteins regulated. What do I mean by that? So cyclins are a protein. They activate cyclin-dependent kinases, CDKs. And once they activate that, that'll trigger a phosphorylation cascade, as we'll talk about in a second on the right side. Now, the nice thing is we only need to regulate the concentration of cyclin because if cyclin is missing, then CDK is inactive. And so it doesn't matter what the concentration of CDK is because in reality, CDK can be something like this all the time. And it's only going to be active when the cyclin that is specific to it is present. So the CDK is only active when the G1 cyclin is there, but when it's not there, even though it has a high concentration of CDK, it's still not active because the cyclin isn't there here. Same thing is true here for the cyclin that is dependent on the CDK that is dependent on the G1S cyclin. It can always have a high concentration, but it's really only going to be active when the cyclin's at its peak. And so we can build up the cyclin and degrade it, and that regulates both. It regulates both the cyclin and the CDK because the CDK is totally dependent on the cyclin. So let's look at how that process works. So let's look at the M cyclin here. The M cyclin binds its specific CDK. The CDK is a kinase, and so what do kinases do? They phosphorylate. That's what I see with these little P's. Okay. And now, hopefully, these are activating these targets, but we're going to see a situation in a minute where it doesn't always have to activate. But it is going to probably activate some target proteins, and these target proteins may be responsible for spindle formation, chromosome condensation, or nuclear membrane breakdown. These are the cell responses, and they're important for triggering the next phase of a cell cycle. These are all important for mitosis, right? Spindle formation is very important to mitosis. Chromosome condensation is very important in prophase, right? And so that's only possible because we have an M cyclin that triggers the activation of CDK, and now the rest of the pathway can go forward. It is very similar to what we've already talked about, but now we're putting a little bit more specific terms with cyclins and CDKs. Okay, now I want to talk about a really cool mechanism for RB. Now, RB is what we call a tumor suppressor. I'm going to write it to the side. I'll try to write it as neatly as I can. I'm using my mouse. Okay, so a tumor suppressor is exactly what it sounds like. It prevents the formation of tumors. How does it do that? Well, we're going to have to really use our skills of figure reading that we've really been practicing this whole unit to understand how it does it. Sometimes I like to look backwards, right? I like to start at the end and then we figure it out from there. So it looks like if I have E2F looking on its own, right? Then I have transcription that drives cell cycle progression. Remember, transcription is gene expression. It's making DNA, it's making RNA from DNA. So, and eventually that RNA will get translated into a protein. We really need to make sure that transcription and translation are nailed down before I move to this test. But if we start at the end, it looks like if I have free E2F, then it is going to transcribe genes that are important to cell cycle progression. So it is going to basically activate the cell cycle. E2F is going to activate the cell cycle. Well, how did we get there? Well, now I'm going to go a little bit backwards. It looks like I have a cyclin and a CDK. If the cyclin is bound to the CDK, then the CDK is active. And CDKs are kinases phosphorylate. So what is this kinase phosphorylating? Well, I'm going to look for the P's. The P's are going to tell me what is getting phosphorylated. So I see P's here. These are for phosphate groups. So it looks like the kinase is actually acting on RB, which is a tumor suppressor. It also looks like when RB is phosphorylated, it lets go of E2F. And so E2F now is free to go drive the cell cycle. That's how it works. Now, why do we call it a tumor suppressor? Well, because as long as RB is bound to E2F, the cell cycle doesn't proceed. But if E2F is on its own, then the cell cycle does proceed. So the tumor suppressor, remember that cancer is related to this idea of repeated cell division that is uncontrolled. And so if there's a problem with E2F or RB, more specifically, if there's a problem with RB where it can't bind E2F anymore, then you really likely will have cancer. And that is the case. This is actually stands for retinoblastoma protein. And so mutated forms of this are linked to eye cancer, retinoblastoma. Okay, so that's what I was leading you here. Make sure that you pause and you understand how, if a protein is mutated, what could happen. Now, I talked about a situation when we were using cancer. That would be the problem if RB was mutated. But you could also potentially have apoptosis. And this is kind of, I don't want to say it's the more preferred scenario, but it certainly prevents the formation of cancer. So you can have normal cell division, but then suddenly you have one that has a damage. Now, hopefully at one of those checkpoints, the cell catches it and it says, oh, if we replicate or if we duplicate, we're going to have a problem, and that could be really bad for my organism. So we go through apoptosis. But in the case of cancer, for some reason the damage is it doesn't get detected, and so it'll proliferate. And if there is a mutation regulating the cell cycle to make it go faster and faster than a normal cell, that's where you're going to typically get very severe cancer and a tumor. So hopefully you now understand how the cell cycle is regulated and how that can lead to either apoptosis or cancer. I want you to spend some time making sure that you can answer these questions in front of you. I'll briefly go over them in a second, but I'd really like you to pause and make sure you understand it first. But transformation is this idea of cells that are essentially unable to die. And so it's usually not in an organism, it'd be like in cell culture. How do cancer cells usually die? Well, hopefully apoptosis catches it before they even become true cancer and true tumors. Hopefully they're caught by apoptosis. Then we actually have some immune system abilities. We're getting some really awesome treatments, but we will get there when we get there. Now, what is the difference between a benign and malignant tumor? Well, these you can just borrow from the English language. Benign means unremarkable. And so benign tumors are typically ones that don't cause severe harm to an organism. I don't want to say that they aren't bad, because they can cause some problems. They can cause like chafing and stuff, which is not ideal. But they're generally not malignant, which means that malignant tumors are the ones that are likely to spread and cause severe problems. So the last one here, I really want to spend some time thinking about it. You might need to, if you don't have the book, go look up the Her protein and really think through how this might work. I'm not going to answer it for you because I really want you to practice on your own. Have fun studying, and I'll see you at the test.